Physical Methods of Microbial Control, Chapter 9. The learning objectives for this topic are, and there's quite a few of them, discuss the advantages and disadvantages of using moist heat in an autoclave and dry heat in an oven for sterilization. Explain the use of Bacillus stereothermophilus endospores in sterilization techniques. Explain the importance of pasteurization and describe 3 different pasteurization methods. Describe the use and importance of refrigeration and freezing in limiting microbial growth. Compare and contrast desiccation and lyophilization. Describe the use of filters for disinfection and sterilization. Discuss the use of hypertonic solutions in microbial control. Differentiate ionizing radiation from non-ionizing radiation as they relate to microbial control. Describe 4 levels of biosafety and give examples of microbes handled at each level. So in this topic we're going to talk about heat-related methods, refrigeration and freezing, desiccation and lyophilization, filtration, osmotic pressure, radiation, and biosafety levels. So under the category of heat-related methods, which I think of as temperature, I'm going to cover moist heat, dry heat, and cold, which includes refrigeration and freezing. Moist heat can be a sterilization method. You're familiar with moist heat probably in the area of boiling water, if there's a boil order on, or in movies, you boil the water to get the instruments in if you're going to be performing backroom surgery or helping with the birth of a baby. Boiling water actually does not sterilize. It does not kill endospores, and it doesn't kill the endospores of botulism. Therefore, it is not considered a sterilization method. There are 2 definitions for sterilization. One is complete kill of anything, up to and including endospores. The other one is commercial sterilization, which is getting rid of botulism endospores. Generally speaking, if you eat botulism endospores, it's not going to be a problem. You're not going to get botulism from it. But if you're canning and you have botulism endospores surviving boiling and they grow in your canned goods, then you can't get botulism. So you're going, but wait a minute, I've canned before and that's getting rid of botulism endospores. Well, you're right. We're using pressure. Okay? If you add pressure, that ups the temperature, okay? Because when you boil something, the water stays at a certain temperature at sea level, it's 100 degrees C, until the water completely evaporates, and then the temperature of your pot will go up and you'll destroy your pot. But if you add pressure, you can up that temperature up to, in autoclaves, it's 120 degrees C. That's high enough. Okay, you've got the pressure on it, you're keeping the steam in so that the water boils at a higher temperature. So that kills the endospores. This also happens when you're canning in your pressure cooker. Generally speaking, in labs and in hospitals and different places, including dental offices, you will use an autoclave where you have pressurized moist heat, and that's going to kill everything. But how do we prove that everything's been killed? Well, we make use of thermoresistant endospores. Now remember from the previous topic that endospores are right up next to prions in difficulty in killing them. And if we take endospores from organisms that are thermophiles, then they're going to be even more resistant. So we're taking the very hardest things to kill and we're testing our autoclave with it. So here's an example of how we would do that. So we have a vial that has another vial full of media, and we have a strip that contains a known amount of endospores, usually 1 million. So we run this through the autoclave. Then after we autoclave our stuff, we let this cool off, and we squeeze this to break the vial of media so that the endospores are exposed to food and fluid. Now, if they're not dead, they're going to wake up, they're going to start metabolizing, and we track this by the use of a pH indicator. If the pH indicator turns yellow, it means that acids have been produced, metabolism has occurred, and you need to get your autoclave fixed. If all of the endospores are dead, then the medium stays red and it stays clear instead of turbid. You also look for turbidity. And this verifies that your autoclave is working effectively. Now, we also use moist heat in when we pasteurize milk. We're not aiming to sterilize milk when we pasteurize it. We just want to get rid of the microbes that are going to cause disease and lead to increased spoiling. Most of these organisms, with the exception of botulism and some of these others, are sensitive to heat. So you don't have to get it up to as high a pressure as you would in an autoclave or even boiling. But this gets rid of most of your microbes. The milk stays fresh for longer, and you don't have to worry about botulism because we don't keep milk in airtight containers. I should explain here that the organism that causes botulism only grows in airless conditions, and it uses endospores to survive situations in which there's air. So that's why I keep saying you don't have to worry about botulism in containers that have airflow. But pasteurization also gets rid of nasties like listeria. That's been in the news with the cantaloupes. Also E. coli and other things that are found in the guts of cows. And not to be gross or anything, but whatever comes out of the end of a cow is going to get on the udder. Now, you're probably wondering why, having heard this, there's been a push lately toward promoting the health benefits of drinking raw milk. Well, when you pasteurize, you do lose some of the nutritional value of milk, because when you're denaturing through heat the proteins of the microbes, you're also denaturing some of the proteins in the enzyme in the cow's milk, and it is less nutritious. And with modern dairy practices, the udder is cleaned very well, and there's been a very low incidence of people becoming sick from drinking raw milk. There have been some, and personally, I'm a microbiologist, and I just can't drink raw milk. Just can't do it. I also can't eat sushi. I've been told it's wonderful, it's tasty, very few people get sick from eating sushi, but I just can't do it. A moist heat sterilization is one of the most common forms of sterilization, especially in the lab or in the clinical environment. But there are times when you can't use it. When you have something that will be destroyed or damaged by moisture. Okay, think, well, in the dental industry, generally it's high carbon steel instruments. High carbon steel instruments are not stainless. They rust really well when they're exposed to liquids. You're probably going, why don't they use stainless steel instruments? Well, they don't maintain their edge very well. Also, if you're trying to sterilize a powder, you really don't want to add water to it. So moist heat is not always the best method to use. So you have to take into consideration with all these methods which one's best for the application. On to dry heat. You've actually been using dry heat as a sterilization method in the lab. Can you think of what it is? That's right, flaming your inoculating loop. We refer to this as incineration. You've heated the wire to the point of where it glows, and you just burn off those poor little microbial bodies. Now, we can do this when we've got a nice thin wire or a thin surface. We also use incineration when we're trying to get rid of large amounts of protein. If a cow is found to have mad cow disease, it's incinerated. So they take it to a pit. In this case, this is probably a picture from when they were getting rid of herds that had hoof and mouth disease. And they would slaughter the animals, put them in a big pit, completely incinerate them, and bury them up. By the way, if they're getting rid of a cow that has mad cow disease, they're not going to allow the smoke to drift off like this, because the agent that causes mad cow disease can survive certain amounts of incineration, but we'll talk about that at the end of this chapter when we talk about how tough some microbes are. In modern society, we use cold all the time to control microbial growth. Cold does not kill microbes. Kills people, generally doesn't kill microbes. So you can put your milk in the refrigerator and it's good for a couple of weeks, and then what happens? That's right, you're on your way to sour cream or yogurt. We generally go ooh when we toss it out. We can also freeze things, and that preserves things for even longer. Now let's talk about why cold does this. Cold is slowing down the activity of molecules so that enzymes function slower and slower. The lipid membrane moves slower and slower, transport in and out of the cell is slower and slower, and it basically puts the bacteria into a state of suspended animation. And if you freeze it, that further enhances it. Now, if you freeze things wrong, then you can kill a certain amount of microbes. Murphy's Law, it's generally a problem when you're trying to preserve the cultures, but if you put a steak that is highly contaminated into the freezer and freeze it for long periods of time, it is not going to reduce the numbers of microbes to acceptable levels. It's just not going to do it. Like I said, it's Murphy's Law. So cold is a control method, but it is not a sterilization method. But it's a very effective one.